Good evening. Yeah, hello. I hope you can all hear me. So, in today's class, we'll look at the sample questions for week 9 of the NPTEL course, Pharmacognosy and Metabolic Engineering. The first question is identify the oxygenated derivative of carotene, which is carotenoid, from the options given below. And four options are given here: zeta carotene, alpha carotene, lycopene, and biolaxanthin. You can think for a couple of minutes about the question and which step requires oxygen, and based on that we'll discuss the answer. You can take a couple of minutes. Okay, first we look at the pathway. So in order for us to be able to produce carotene, it goes back to the precursor, which is a part of glycolysis. It's acetyl-CoA. So from acetyl-CoA, we get malenolonate, and then we get IPP and DMAPP, which we had seen earlier. After which we have gernyl phosphate, which upon the action of enzymes in this particular pathway, which are referred to here as CRT enzymes, CRTE, B, I, Y, Z, and so on, we get lycopene. This is the first precursor that sets the flux towards production of carotene and its derivatives. And shown here is the structure of each of these. Like for example, this aliphatic compound is lycopene, and there is no closure here. It's just linear. Whereas this is a branched, and there are two cyclic rings here. This is beta carotene. Now, after this, in each of these steps, we have ferredoxin O2 involved, but it doesn't get incorporated until this step where we have anther-axanthin, where it is covalently linked to an oxygen here, and it has an OH here. Sorry, zeaxanthin and anther-axanthin. So here you have an OH. This is the first step where the lycopene derivative is having an oxygen involved, right? So they ask identify the oxygen derivative of carotene from the options given below. So if we look at this, we can eliminate lycopene and carotene as well, which leaves us with violaxanthin. And as we observe here, among the three oxygenated derivatives, all are derivatives of beta-cryptoxanthin, only violaxanthin is present in the option. And in general, we have two of these classifications in carotenoids. One is carotenes, that are just the hydrocarbon molecules, which do not have any oxygen. To this belongs lycopene, alpha, beta, zeta carotene. The other is xanthophylls. These are the oxygenated derivatives like zeaxanthin, viola, xanthin, and lutein. So this contains an oxygen in the form of hydroxyl group or epoxyl group. So the epoxyl group is the one here where you have oxygen covalently linked to two carbon molecules, making it an oxygenated derivative. So the right answer is violozanthin. Right. The next question is identify the correct step in the carotenoid biosynthesis that is not catalyzed by lycopene cyclases. So there are four options given here. Trans-lycopene to gamma carotene to alpha carotene, beta carotene to beta cryptoxanthin, gamma carotene to beta carotene. You can take a couple of minutes and then think about the question. We'll then look at the pathway first again, and in which step these lycopene cyclases act, and accordingly decide the answer. Right. So first we look at the pathway here and the steps catalyzed by lycopene cyclases. So we have about three lycopene cyclases, and each of these act on either lycopene or derivatives of lycopene. So lycopene can give either zeta carotene or beta carotene. This is based on whether it's being acted upon by lycopene epsilon cyclase or lycopene beta cyclase. After this, lycopene beta cyclase, it acts on either of these carotenes to give alpha carotene and beta carotene respectively. So structurally what is happening is you can observe that here it's linear in lycopene, whereas in carotene, in both the carotenes, you have a phenyl ring formed. This is the zeta and gamma carotene, whereas alpha and beta have two of these rings on either sides. So this is the reason this enzyme is called as a cyclase. Now, going back to the question, which of these steps is not catalyzed by lycopene cyclases? So lycopene to carotene, gamma carotene to alpha, gamma carotene to beta. These three are catalyzed by lycopene cyclases. But beta carotene to beta cryptoxanthin, which is a step here, beta carotene to beta cryptoxanthin, it's being catalyzed by beta carotene hydroxylase. So that is the step is not catalyzed by lycopene cyclase. So option C is the right answer here. Again, that is mentioned here. The next question is, certain flowers have been noted to contain volatile compounds resulting from the cleavage of carotenoids in their fragrance. Among the following compounds, identify the volatile compound that can be found in such flowers. Option A, beta apo 10' carotenol. Option B, beta lonone. Option C, pyxin. Option D, quercetin glycosides. You can take a couple of minutes to think about the question and then we'll discuss the answer here. Right. So here, if you look at the options, first we look at the other options. One is beta-apotin carotenol. This is an aldehyde, but it is not very volatile. Whereas Bixin is non-volatile pigment, and these glycosides are water-soluble and is responsible for the saffron color. When it comes to beta-lanone, this contributes to fragrance in some flowers, and this is formed by oxidative cleavage of carotenoids by enzymes called as carotenoid cleavage dioxygenases. Now, because the question is about a volatile compound in flowers and resulting from cleavage of carotenoids, beta-lanone is the right answer. The next question is, glandular trichomes share a general structure. Identify the cell type that cannot be found in glandular trichome. Basal cell, companion cell, stalk cell, and secretory cell. So, these glandular trichomes, these are specialized epidermal structures that are found in plants that produce and secrete metabolites. Earlier, we had seen laticifiers and other uh, specialized structures. Again, this glandular trichome contains the basal cell that would anchor the trichome to the epidermis, which will physically link it. Stalk cell that would connect the basal cell to the glandular or secretory cells, and secretory cell that is responsible for producing and storing secondary metabolites. This aside, companion cells, these are part of the phloem tissue and are associated with C tube elements for nutrient transport, and these are not present in glandular trichomes. So here, option B, companion cells, is the one that cannot be found in glandular trichome. The next question is, identify the correct biosynthesis pathway of lutein from phytoene. And four different options are given here. Phytoene gives zeta carotene, which gives lycopene, which gives lutein. Phytoene gives lycopene, which gives zeta carotene, which gives lutein. Phytoene gives gamma carotene, which gives zeta carotene, which gives lutein. And phytoene gives alpha carotene, which gives zeta carotene, which gives lutein. We have seen the pathway of this production of conversion of phytoene to lycopene to carotene earlier. Think about this for a couple of minutes and then we will go back and check the pathway. Yeah, so for this question, we'd earlier seen the production from phytoene to lycopene and how it involves the formation of different carotenes, right? And then finally to lutein. So there are two pathways that we can look at here. One is this one where phytoene is converted to lycopene by CRTI, then lycopene to beta carotene by CRTY. This is the phytoene lycopene carotene pathway. From lycopene, we get either we can get beta carotene or zeta and then alpha and then lutein. So the order is phytoene, lycopene, zeta and then lutein. Option B. Okay, option B. And here we've given what is the specific classification or the chemical step involved. For example, phytoene, it's the precursor in carotenoid biosynthesis. Lycopene, it's formed by desaturation of phytoene. Zeta carotene, it is the intermediate in the pathway. And lutein, it is a xanthophyll, meaning it has um, something other than carbons and hydrogen. Specifically, lutein here has oxygen. Okay, so this is how lutein is being formed. The next question is identify the pharmacological property for which the compound artemisinin is used as a drug. Option A, hepatoprotective. Option B, anti-cancerous. Option C, anti-malarial. And option D, anti-diabetic. You can take a couple of minutes to think about this question. Okay, so this is a very well-known uh, application of metabolic engineering and synthetic biology. Artemisinin was famous because of its role in malaria treatment. So this drug is derived from the plant Artemisia annua, and is particularly effective against Plasmodium falciparum. Right. The next question is in vitro shoot cultures of Hypericum perforatum upon microscopic examination revealed translucent glands in leaf lamina, which is the site of accumulation of a bioactive compound. Identify the correct compound. Option A, hypericin. Option B, hyperforin. Option C, hyperforin. Option D, add hyperforin. Right. So this species called Hypericum perforatum, it's also called as St. John's wort. Now this produces two of the compounds that are listed here. One is hyperforin, the other is hypericin. Uh, while hyperforin is the compound that is specifically accumulating in translucent glands of the leaf lamina, hypericin is found in the dark colored glands and not the translucent glands. That's the difference. And specifically, hyperforin is a secondary metabolite that has been um, used as an antidepressant, antibacterial, and anti-inflammatory. And it has anti-inflammatory properties as well. The next question is, red color of the tomato is caused due to accumulation of dash that is synthesized inside dash from the isoprene precursor dash. So you might be familiar with this, but still we'll look at the options. Option A, violozanthin, vacuole, geranyl pyrophosphate. Option B, lycopene, plastid, farnesyl pyrophosphate. Option C, lycopene, vacuole, geranyl pyrophosphate. And option D, lycopene, plastid, geranyl pyrophosphate. You can take a couple of minutes and think about this question. Right. So first, the color is due to accumulation of lycopene. Now we need to know where lycopene is synthesized and what is the precursor from which it's being synthesized. If we look at the pathway for lycopene synthesis, either this one where we see geranyl pyrophosphate, or even this one where we have geranyl pyrophosphate. So that is the isoprene precursor, although that itself is being derived from farnesyl pyrophosphate. So it is synthesized in plastids, chloroplasts, and chromoplasts, where carotenoid biosynthesis occurs. The isoprene precursor is geranyl pyrophosphate. Right. The next question is, autooxidation of artemisinic acid gives rise to option A, artenoin B, option B, artemisinin, option C, dihydroartemisinic acid, and option D, artemisinic alcohol. We look at the pathway for this question. You can take a couple of minutes to think about the question. Oh, hi Swati. So I have gone through the questions. I can repeat them so that you can benefit from it. I hope you can hear me. Can you hear me?
Yes, sir.
Okay, sure. So we'll quickly go through the questions. I was just going through the questions for I think I was in question 7 or 8. Yeah. So the first question is identify the oxygenated derivative of carotene. And the options given below: zeta carotene, alpha carotene, lycopene, violazanthin. Majorly, these are hydrocarbons. After a particular step, they have addition of oxygen. You can think about this and then let me know. We have the pathway with the structures. We can look at that and finalize the answer. Right. So, do you want to try telling the answer here for this question?
You are asking precursor or
No, no. So the derivative of carotene, which has an oxygen. So if you see all these compounds, once journal pyrophosphate gets converted and it forms phytoene and so on, you only have carbons and hydrogens in it. Now the question here is asking about oxygenated derivative. So which of these has an oxygen in it? That's the question. So if you know the structure of few of these compounds, you'll be able to more or less guess it. Also, these in carotenes, you have two classifications: carotenoids and xanthophylls. While carotenoids have only carbons and hydrogen, xanthophylls have both caroten. In addition to carbons and hydrogens, it has oxygen as well. So, which of these has an oxygen? That's the question.
Uh, is it while option D,
Yeah,
viola xanthin?
yeah. You're right about that. So, if you see lycopene, all carotenes, they have only carbons and hydrogens, although the degree of saturation and saturation would vary and the cyclization would vary. Valexanthin is the one that has oxygen, and this starts with this particular step after beta cryptoxanthin, which gives you zeaxanthin and anthraxanthin and valexanthin. All of these has an hydroxyl group here, and like I mentioned here, um, carotenoids are divided into two classes: carotenes and xanthophylls. While carotenes are purely hydrocarbon molecules, xanthophylls have the oxygenated derivatives. Right. The next question is identify the correct step in the carotenoid biosynthesis that is not catalyzed by lycopene cyclases. So, if you look at this pathway till lycopene, it's all linear. None of these are cyclized, although you have branches. But once you have lycopene cyclases, you would start getting cyclic molecules at different ends, based on which you either have the alpha or beta or the gamma for the carotenes here. So now the question is, which of these steps doesn't require lycopene cyclase? Is it option A, trans lycopene to gamma carotene, or option B, gamma carotene to alpha carotene, or option C, beta carotene to beta cryptoxanthin, or option D, gamma carotene to beta carotene? Yeah, so do you like to try answering this question?
Is it option C? I'm not sure.
Yeah, it's option C, beta carotene to beta cryptoxanthin. So if we look at the role of different lycopene cyclases, from normal lycopene, we either get gamma or delta or gamma carotene, and you have two cyclases, epsilon cyclase and beta cyclase. So what this does is at either extremes, it forms the cyclic moieties, right? Then this beta cyclase itself would convert this delta and gamma into alpha and beta carotene respectively, where in the other end you have a cyclic group. So now you have from a linear lycopene, you have one cyclic group. In the second step, you have two cyclic groups. Then you have, so this is the part involving lycopene cyclases. Whereas this step, beta carotene to beta cryptoxanthin, that is taken care of by this enzyme called beta carotene hydroxylase. So that is the step that doesn't involve lycopene cyclase, which is the question that's being asked. It's option C, beta carotene to beta cryptoxanthin. Right. The next question is, certain flowers have been noted to contain volatile compounds resulting from the cleavage of carotenoids in their fragrance. Among the following compounds, identify the volatile compound that can be found in such plants. Um, Option A, beta apo 10 prime carotenol. Option B, beta lonol. Option C, pyxin. And option D, quercetin glycosides. So for this question, you can first think about uh, which of these compounds are actually volatile. So if they have to be volatile, their solubility has to be less. So think in terms of that, you'll be able to eliminate one or two options, and then we'll take it from there. Yeah, so from the name itself, do you think any of these will be more water soluble and will be non-volatile? Just from the name of the compounds given here, any idea? We didn't discuss about specific compounds, but we discussed about a specific modification in other classes of compounds. that makes it less volatile and more water soluble.
Is it Bixen?
Bixin, I'm okay. Just from the name. Okay. I was mentioning about glycosides. So glycosides are okay. We are trying to okay. Bixin. Are you telling that Bixin is the right answer, or you like is it a part of the elimination?
Mm, um, the volatile among the following volatile compound.
So we can eliminate just by the name. If you see here, there is something called as glycosides. Glycosides means these compounds to which glucose is covalently attached. So they become more water soluble. And if they're more water soluble, they'll become lesser volatile. So you can eliminate this quercetin glycoside with that itself because it's not volatile anymore. I was just trying to give you an idea on how to eliminate the wrong option here. That's why I asked that. Bixin again, it is non-volatile and it's a carotenoid pigment. So we've eliminated Bixin and quercetin glycosides. So now we are left with beta-apotin carotenol and beta-lonone. Do you want to try commenting on these?
Is it option A?
Option A again, it's not very volatile. So and it is not formed by cleavage of carotenoids. Whereas this beta-lonone is the one that is formed by oxidative cleavage and by specific enzymes like for example here carotenoid cleavage dioxygenases. So that's why beta-lonone is specific here. If they had just talked about volatility, you could have mentioned this beta-apotin prime carotenol, but because you're mentioning about the oxidative, the cleavage, we'll have to go with beta-lonone. Again, these are I know it's very difficult to remember all these informations because of these too many pathways. But one way to try to answer these questions is through elimination because most of these are not directly embedded. You can't remember all of these. Right. The next question again. Okay. So this is about vascular biology. So um, in plants. So glandular trichomes share a general structure. Identify the cell type that cannot be found in glandular trichome. Option A, basal cell. Option B, companion cell. Option C, stalk cell. And option D, secretory cell. So you can just think about how we talked about sieve elements, sieve tubes, companion cells, and where it is present, xylem, phloem, uh, and so on. What we had discussed earlier. Also about laticifiers when we discussed about rubber, latex, and plant-based products. So think about it, and again, if you're able to eliminate that one option that you had earlier seen from the previous classes, you'll be able to get the right answer here.
Is it basal cells?
Yeah, can you hear me? Yeah, yeah,
Yeah. It is identify the cell that cannot be found.
yeah, yeah,
Question
yeah,
or
yeah, yeah. The cell that cannot be found. So earlier we saw the uh, role of a particular kind of cell when it came to xylem and phloem. We talked about sieve elements, latissifiers, specifically in that uh, portion, I think week 2 and week 3, where we also talked about sieve elements, sieve tubes, and from where to where particular biomolecules would flow, particular polymers would flow. So, does any of these options remind you of that?
is it cannot be found secretory cells?
Oh, no. Okay, so earlier when we discussed about sieve elements and sieve tubes, we had also talked about companion cells. So this is a part of the phloem tissue that's associated with sieve tube elements. Uh, so they are specific for nutrient transport. That's why they cannot be a part of this glandular trichome. Whereas the secretory cell is responsible for producing and storing secondary metabolites. And the stalk cell, this connects the basal cell to the secretory cell. So it's like you have basal cell, which would anchor this trichome, meaning three different to the epidermis. So the epidermis is there, and you have the basal cell, and the stalk cell, and then the secretory cell. The stalk cell is in between basal cell and the secretory cell, and all of these are connected to the epidermis. And here, companion cell has no role. Was that clear?
Yeah. Okay. Yes, sir.
Okay, fine. This is a simple, straightforward question. I think you might be familiar with this. So here you need to identify the correct biosynthesis pathway of lutein from phytoene. So we know from phytoene you get lycopene and then lutein. So you need to determine the order here. And earlier also we saw one pathway which showed how lycopene cyclases act to produce different carotenes, the gamma, the delta. the alpha, beta carotenes, and then the lycopene, and then lutein. So you can think about the specific options here and uh, let me know which one you think could be the possible uh, pathway for this product formation. So this alpha, beta, gamma, delta, zeta, all of these are based on the region where the cyclization happens. So you just need to remember that. We can go through the structure once again so that you'll become familiar. Yeah, so once you finalize your option, you can let me know.
Is it option C?
Option
Actually,
C.
what is
So we need to find the pathway through which phytoene is getting converted to lutein. So you need to think which is the first two lycopenes or which is the first set of carotenes that are formed from lycopene. Remember, like earlier we saw here, from lycopene we have delta and gamma. And then from these two carotenes we have alpha and beta respectively. Now to get lutein, we need to focus on alpha carotene. So reverse back the pathway, you will get lutein from alpha carotene, from delta, from lycopene.
then? Is it option C is correct?
Option C. So here, 1 minute. So we have phytoene. From phytoene, do we directly get these two? Uh, Let me check. From phytoene, we get lycopene. From lycopene, we get this carotene. Do we get gamma and then zeta carotene? Or how does it work? I did look up on this and I couldn't find this zeta, zeta carotene. So what I could think is, because we are not sure about what this means, whether it's beta or zeta or the thing, the safer option that I had chosen was phytoene to lycopene to zeta carotene to lutein. But again, from gamma we get only beta carotene. So if you're talking about gamma carotene, you will not have the production of lutein, because for lutein we need alpha carotene. If you see here, alpha carotene is the only way through which lutein is being produced. Like we saw earlier, lycopene, upon the action of different cyclases, either produces delta carotene and gamma carotene. Here, delta carotene produces alpha carotene. Only that would give you lutein. Gamma carotene produces beta carotene. That will never produce lutein. So, based on that, you should be able to say whether gamma will be helpful or not. Uh, are you getting a hint of the question?
Yeah, I got it that alpha carotene is required for lutein.
Yeah, alpha is only required, and if alpha has to be produced, you need delta and not gamma. If gamma is here, then it will not give you alpha carotene here, which is why this option might not be right. Does it make sense?
So is alpha carotene is not there in anyone.
No, no, alpha carotene is here, but you can. Alpha carotene is here, but again, this option of zeta carotene, I am still not sure, which is why I thought this could possibly be the right option. We know
Okay,
phytoene produces lycopene, and from lycopene we get lutein.
okay.
So this seemed like the most appropriate answer from here.
Sir, just
Ha, sure, sure.
one question was there in assignment about the promoters.
Okay,
But in the lecture, it was, I think it was not taught or I missed it. The promoter, that
okay.
AP1 and GL2, all that, can you just give us some idea? Because it is not mentioned or I missed somewhere.
Promoters. I'll check on that, and maybe in the next class I can.
That
Which
AP1, it was a
week's
question
assignment question was this? Week 8, is it?
this week, ninth one.
Ninth
That
week.
some, yeah, AP1, then GL2, all those promoters was asked.
What was the question?
Question, I have to check.
Okay, you can maybe check, and you can even post it in that group. I think there is one Google group for this course, NPTEL Pharmacognosy and Metabolic Engineering. You can posted question there. That's how we operated in the previous semesters for the NPTEL courses as well. We can check and we can let you know that.
Okay.
Sure. Yeah, so that's how that question would end. Next is a very straightforward question. Which pharmacological property does artemisinin have? And we
It
know
is
that,
option C,
uh,
anti-Malarian.
yeah, we know that it's antimalarial. Straightforward question. And after that, we have this question. In vitro shoot cultures of Hypericum perforatum revealed translucent glands in the leaf lamina, which is the site of accumulation of bioactive compound. So identify the correct compound. And four options are given here. Hypericin, hyperforin, secohyperforin, and adhyperforin.
Option B, hyperforin.
Yeah. What about hypericin? That is right. Hyperforin is right. Hyperforin is also produced in the same plant, Hypericum perforatum. So Hypericum perforatum, it accumulates in the translucent glands, whereas hyperforin, it accumulates in the dark colored glands. That's the only difference.
Hypericin. Okay,
Right.
hypericin is found in dark colored glands. Okay.
Glands, yeah. Both are produced in Hypericum perforatum, but because the question specifically asks for translucent glands, that's why we go with hyperforin. Right. The next question is, the red color of the tomato is caused due to accumulation of dash, that is synthesized inside dash from the isoprene precursor dash. Again, so this again is a straightforward question for at least the first two options. So, do you know which organelle is responsible for pigment production? There are specific sets of organelles within cells that are essential for pigment production. Any idea about that?
Um, the red color of tomato is caused due to accumulation of lycopene.
Lycopene. Okay, so now we are up to three options.
Synthesized inside plastid
Plastids.
vacuole.
Right. Yeah, plastids are these organelles. So you have variations in plastids depending on the pigment that's being accumulated. One specific organelle or one specific variation is chloroplasts, where chlorophyll is synthesized, that we are familiar with. And because lycopene is also causing pigmentation, that's also produced in plastids. Right. Now, now that you told plastid and lycopene, do you think both farnesyl pyrophosphate and geranyl pyrophosphate are applicable for this question?
Is it geranyl pyrophosphate?
Right, that's the answer. But where do you think geranyl pyrophosphate is being produced from? Any idea?
geranyl pyrophosphate.
It's produced from farnesyl pyrophosphate. So technically both are valid, but this is the immediate precursor. So we would go with option D. Right. The next question is auto-oxidation of artemisinic acid gives rise to option A, artenoin B, option B, artemisinin, option C, dihydroartemisinic acid, and option D, artemisinic alcohol.
It is option B, autooxidation, artemisinin.
Artemisinin. Okay. Do you know the pathway through which this is being produced? Different alcohols to aldehyde to acid to dihydroacid and then artemisinin. Do you know this pathway? The answer is right.
The structures I don't remember, but
Okay, okay.
yes, aldehyde, yeah,
Alcohol to aldehyde to acid.
aldehyde to acid, yes.
Yeah. From artemisinic acid, again it gets reduced to dihydroartemisinic acid. Then it gets this step, it happens to give you an oxygen here in the ring region, which gives you artemisinin, right? And requires singlet oxygen or superoxide radicals. Okay. The next question is to create. Oh, yeah. Oh, we've come to the end. Huh. To create golden rice, one plant gene and one bacterial gene were transferred in rice. Identify the correct genes and their respective sources. Okay, sources are bacterial and plant, and the gene names are given here: desaturase and synthase. So there's also a prime reason as to why a particular bacterial source was used, which we'll come to once you have finalized your option. Hmm. Yeah, you can try answering.
Uh, PSY and CRTI.
Okay. Any idea why CRTI? Oh, okay. PSY and CRTI, that is fine. Uh, and the source?
What you asked, sir?
No, no. So, do you know why bacterial CRTA was used? The answer is right. Answer is option B. Plant phytoene synthase and bacterial desaturase, the CRTI. Any idea why that is specifically used? Because it's a plant, they could have specifically used plant genes itself, but instead they used one plant gene and one bacterial gene. I'm asking, do you know
To,
why one bacterial gene was specifically used?
to increase the level, high carotene
That you would get by using the
to
plant gene
achieve.
also, right?
Okay. Is it endosperm specific promoter gene or
No.
it's endosperm
So
specific?
this gene, the product of this gene helps in skipping a number of steps. So this is a bacterial transformation process. Chemically, something is being modified by the enzyme. Usually, for this process, it takes a number of steps. Yeah, usually it takes a number of steps here. Uh, like you see here. But usage of this bacterial gene from this organism, Erwinia iridovora, that bypasses this multiple desaturases. That is why this bacterial gene is used instead of the plant gene. And of course, the plant gene it's taken from maize. There's also a specific paper that talks about that uh, how maize had come to the rescue for producing golden rice. You can check that paper that goes into detail of how this maize gene was utilized. Right. So the answer is right. You just need to know the reason why we use this bacterial gene. Huh. So we've covered these questions. If you have any doubts, you can ask me here. And for that specific question, you can post it in the Google groups, and I'll look at the question and I'll reply that there. Okay.
So can you some few questions, first few questions, can you just show?
Huh. Sure. Yeah. So this is the first question, which talks about the oxygenated derivative.
This question I was there. This is the first question.
Yeah. Yeah. This is the first question.
Oxygenated that violazanthin.
Yes. Yes.
Yeah, I was there.
Yeah.
Okay. Okay,
So
sir.
we've actually
Thank you.
covered all the questions. Yeah.
Okay. Okay.
Sure. Thank you.
Thank you, sir.
Next class we'll have on Monday.
Okay. Thank you, sir.
Yes. 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 Thank you. You can leave. Sindhu, you can also leave. The session is over unless you have doubts.